Students are always saying to me they really struggle with application skills and how to take the knowledge that they have and apply it to the question that's been given to them. So I thought I would do a revision looking at skeletal muscle cells and looking at respiration. So you're told in this question that skeletal muscle cells were broken up and separated into parts using centrifugation. Now, each part was then mixed with either glucose or pyruvic acid. Now, the parts that were tested were the mitochondria with cyanide, and then the rest of the cell content, the cytoplasm. And what the scientists were checking for was to see if there was a production of carbon dioxide or a production of lactate in each of the different samples. Now, the results table has been given to you. So the first thing I would say to you is the fact that we have been told in the question that we're looking at respiration because they have mentioned the mitochondria. So we know we've got respiration. Also, we need to add in, if we're looking at mitochondria, we are looking at aerobic respiration because that is where the mitochondria is involved. Now, there is another type of respiration, remember, and that is anaerobic respiration. And anaerobic respiration is where we're going to have the production of lactate. So this is really important, us just bringing that knowledge to the forefront of our mind. So now what we're going to do is we're going to look at the information they've given us and try to decipher what they're telling us to explain the results. Now, the first thing I would always do, again, just reminding myself, is what do I know about respiration? So I know I've got glucose. Glucose is broken down into pyruvate through the process of glycolysis. I know this, and I know that this happens in the cytoplasm. So I'm just giving myself little hints. The pyruvate is then moved into the mitochondrial matrix. So we're now in the matrix. It is now going to be changed in the link reaction to acetyl coenzyme A. Now, acetyl coenzyme A can then be taken and used in the Krebs cycle. Again, the Krebs cycle, we're looking at the matrix inside the mitochondria. Now, that's just my little hint to myself. And then I'm going to add in a bit more because they've actually said in the question about the production of CO2. Now, I know that glucose being broken down into pyruvate does not produce any carbon dioxide. However, I do know that in the link reaction, I produce CO2. I also know that in the Krebs cycle, I produce CO2. So therefore, my expectation of these results is that CO2 will be produced where there is the mitochondria. So let's just look at what they've shown us then. So in this question, we'll start with the cytoplasm. So there is no carbon dioxide in the cytoplasm being produced because it's showing us as it being absent. Well, that's what we expected because remember, the cytoplasm is where glycolysis occurs and therefore we're not expecting there to be any carbon dioxide. However, when there is lactate being produced in the cytoplasm, that tells us that the anaerobic part of respiration has to be occurring in the cytoplasm. So therefore, I know that if there is no oxygen present, the pyruvate can be converted into lactate so that we can regenerate. The reason we do that is so that we can regenerate our NAD coenzyme and therefore allow for glycolysis to happen again. So again, that is an expectation that we would have the lactate produced in the cytoplasm if we are doing anaerobic respiration. Let's look at what happens if we have incubation this time of pyruvate in the cytoplasm. So we're now looking at pyruvate, and we know that the pyruvate needs to go into the mitochondrial matrix for the pyruvate to be converted into the acetyl coenzyme A? Well, we can see that carbon dioxide is absent when there is pyruvate incubated in the cytoplasm because there is no way in which we can convert the pyruvate into acetyl coenzyme A without the enzymes that are needed within the matrix. So that shows us that doesn't happen. The other thing for us to note is that in the cytoplasmic residue, the lactate is actually not being produced. So here's a question then. Why is it that the lactate isn't being produced in the cytoplasm if we know that pyruvate can be turned into lactate to regenerate the NAD? And this is the interesting part. You should know that the NAD, which is NAD plus, is going to be converted into reduced NAD through the process of glycolysis. The pyruvate being converted to the lactate would take the reduced NAD back to being the oxidized NAD plus. Well, we didn't have any glucose. Therefore, there was no reduced NAD being produced. So there's no hydrogens that can be donated to the pyruvate to turn it into lactate. So that is why it is absent in the cytoplasmic reticulum. Now let's look at the mitochondria then. So what can we see with the mitochondria? If it's incubated with glucose, then there is no carbon dioxide produced. That's because the glucose needs to be hydrolyzed down into the intermediate products through the process of glycolysis. So if there's no glycolysis happening, because we're already inside the mitochondria, we cannot metabolize the glucose into the necessary molecules. So that's why it's absent. And obviously, again, we have the absence of the lactate because the glucose hasn't been metabolized. When we look at it with regards to the incubated with pyruvate in the mitochondria, we can see that carbon dioxide is being produced. And we know that carbon dioxide is produced in the link reaction, and we also know that carbon dioxide is produced in the Krebs cycle. So that is an expectation that we would expect that to be present. And obviously with the pyruvate, the lactate isn't actually present, and that's because again, we don't have the NAD being reduced and not able to go to oxidative phosphorylation. So we can expect that there is oxygen present, and therefore that is able in the mitochondria to go to the cristae for oxidative phosphorylation. So we wouldn't expect lactate to be produced because we can have oxidative phosphorylation. So let's look at the mitochondria with the cyanide then. So mitochondria with cyanide, we can see with the glucose, just like with the mitochondria on its own, is exactly the same. So with the cyanide, it doesn't change anything because we're wanting to see what's happening in the mitochondria. So if we compare the mitochondria with cyanide, what we can see is the big difference is that carbon dioxide is not being produced. And the reason the carbon dioxide is not being produced, even though the mitochondria is being exposed to pyruvate, is the cyanide must be inhibiting the reactions from occurring. So whenever you get a question like this, which is an application question, it's really assessing your knowledge of the topic area, in this case, respiration, hidden as if it's to do with the skeletal muscle, but actually it's really looking at, do you know the difference between respiration and the aerobic and anaerobic respiration and where it occurs? And also, do you understand that respiration is a controlled metabolic pathway which requires enzymes, and therefore if we put in any molecules that are inhibitors, we can inhibit the process from occurring.